Hi everyone, I'm Olivia Sargent, your host of Science Minds. Today we're in the Purple Tree Cafe at the University of California Davis Mind Institute, and we're joined by Dr. Elizabeth Berry-Kravis from the Rush University Medical Center. Welcome, Dr. Berry-Kravis. We're so excited to have you.
Thank you. I'm really glad to be here.
Fabulous. Well, I'm wondering if we can just get started by getting a broad sense of the type of research that you do, the questions that you ask, the populations that you work with.
So I do research in neurodevelopmental disorders. I'm a pediatric neurologist by training, and I really got my start in research in the Fragile X field, which I have been studying since 1988. So for a very long time. And as time has gone on in working with the Fragile X population based on research coming out, we've done a lot of work on translational research. So we've done we do some work in the basic science lab, although mostly it's kind of applied research trying to understand how like genotypes or the amount of Fragile X protein impacts the patient's clinical symptoms. But also we've done a lot of work trying to translate treatments that are discovered, like to help the Fragile X mouse or to work in Fragile X cell lines or something like that. How do we move those treatments to potentially treat humans with Fragile X based on targeting a mechanism that's related to their disease, as opposed to just using like a supportive treatment like seizure medicines or behavior medicines. We've also spent a lot of time studying Fragile X, the manifestations of Fragile X and how to best treat the condition through natural history, and have spent a lot of time developing outcome measures that will measure things that are important in Fragile X to show whether like a drug is working in a trial.
Wow, so it sounds like you have a very diverse kind of range of research interests and focuses, but a lot of it sort of ties back to this Fragile X community, right?
So because we've actually branched out now in probably the last 10 years into Rett syndrome and Angelman syndrome and many other neurodevelopmental disorders that are working toward having treatments that target their underlying disorder as well. And so the work overlaps a lot for different syndromes in terms of the kinds of things that you have to do.
So I'm curious sort of how you got your start in this field. You said you've been working in this, you've been doing this line of work for 40 years, right? 30?
Not quite.
Almost 40
but,
years?
Something close to that.
How did you first get into it?
Well, I will say that in 7th grade I read a book that was called Death Be Not Proud, that was about a boy with a brain tumor, and at that time I decided I was going to become either a neurologist or a neurosurgeon, and would definitely go into neuroscience. And so I pursued that, throughout high school and college, went to medical school, and then decided to go into neurology, and specifically pediatric neurology, because I really liked working with the kids, actually. And so then really the Fragile X work came out of, I've always been interested in how do neurons communicate with each other, and that's what a lot of my research, like in my undergrad and stuff, was on. So I've wanted to know, like I would sit in 7th grade and think about, I've got all these neurons in my brain and I'm thinking how to do this math problem. And it's based on those neurons firing that I'm able to think that, and I really would like to know what's going on in those neurons to understand how they, quote, think. So as a result, I studied a lot of signaling pathways and receptors that have to do with neural communication early on in my PhD days. And then I got into the Fragile X field because we wanted to study how a certain pathway in the brain, which also is present in other cells, like platelets, for instance, works. And so we studied a disease called pseudohypoparathyroidism because patients with that disease had a defect in this system, which is called the cyclic AMP system. And we had Fragile X patients as controls because we wanted a control group with intellectual disability. And it turned out the Fragile X patients had a bigger defect in this system than the patients we were studying. And so we went on to study that in Fragile X. And in the process, I met a lot of wonderful families who came in to give blood to my project, and they thought they needed a Fragile X clinic in Chicago because they had heard that Rondi Hagerman had a clinic in Denver and they wanted to have one in Chicago. And they had been trying to get several people to run a clinic and nobody could set it up. So I set up a Fragile X clinic in 1991, and then we've been running ever since. And now we have a lot. We've seen 700 to 800 patients, and we see a lot of them regularly, and so it's been a program ever since then. And once I had the clinic, then it was just natural to move into all this translational research, because the translational research in Fragile X has really been directed toward what I wanted to understand to begin with, because Fragile X is basically a synaptopathy. It's a disorder of synapses or brain connections where the connections aren't forming right, and so you can't learn as well. So it's a window into how does the brain work to make those connections, which circles back to what I was thinking about in seventh grade.
So your seventh grade self
So
would be
really
very pleased. It
yes,
sounds.
it's been the ideal field for me to work with, and it's just helped me expand into other disorders that are also synaptopathies, like Angelman syndrome and Phelan-McDermott syndrome and a bunch of other developmental disorders. So it's been really a very fulfilling field that I may never retire from because it just keeps being more interesting. But
that's so amazing. What a great scientific journey to go through. And I love that it was your control group that
Yes.
originally got you into this work. That's, yeah, just sort of like one of those happy scientific accidents, right? That leads
It was,
you.
I would say it was a scientific accident.
Yeah.
Some of the best things have been discovered by scientific serendipity, though.
Yes, yes, exactly. That's amazing. You mentioned too that you have both this clinical training, right? You have an MD, and then you also have a PhD in biochemistry, right?
Right.
And I'm wondering how your research sort of merges across these clinical and kind of basic research fields. It seems like it must give you such a unique perspective that situates you to do the work that you're doing.
Yeah, my lab has continued to do some, sort of basic, mainly on like cells and proteins that are regulated by the Fragile X protein in different kinds of cells. We've also, because we needed to understand the Fragile X mutation and how it impacted the clinical situation, we've expanded to doing a lot of molecular biology, like the repeat size of the mutation and the methylation, and then we've learned to measure the Fragile X protein. And so we've done. I would say that we don't do basic science. I think anyone who does as much clinical work and translational work, like clinical trial work, that as I do, there's like no way for you to be running a basic science lab as well. But we do a lot of collaboration where we may get samples and process samples, and then send them to other sites that are doing basic science research, or we may be running an assay on, for instance, if there are a whole bunch of patients with a certain condition that may relate to Fragile X or may not, we will run a bunch of a genetic assay, or if we need to run the Fragile X protein. So we'll run an assay on a whole cohort of patients for somebody else who's studying something purely clinically, so we can help them with the molecular parameters that might be predicting, for instance, clinical phenotypes or clinical responses. And then we've done a lot of work on biomarkers for clinical trials, like what can you measure in a person with Fragile X's blood or whatever that would predict a response in clinical trials. And sometimes from doing biomarker work you discover things. Like last year, 2 years ago I guess, we were doing a project with Joel Richter's lab on RNA profiles that might be biomarkers, and we actually discovered that there's a situation where the Fragile X gene is making certain kinds of RNA in certain patients, and we could actually treat that problem with an antisense oligonucleotide
That's a big word.
right, to allow more of the Fragile X protein to be made and potentially fix the cells. So this is now a possible treatment for some of the patients with Fragile X in the future that we're continuing to evaluate to determine if it will work. So I think in that way we've been very collaborative, and I think the clinical work just meshes into the basic science work, not just in our lab, but in all of the places where we collaborate. And of course, we've become, over time, because we started in clinical trials very early when there were first some mechanisms that were thought to be targetable by new medications that might work on the actual Fragile X, we've been doing clinical trials for many years, and so we have a very large clinical trials program as well. But all of this work is interconnected, because you can't do clinical trials and understand how to measure change in people with Fragile X unless you understand people with Fragile X and what their problems are very well. So the trials won't be successful unless you can understand the clinical work. And you may not be able to measure what you want to measure in the trials unless you develop it, within the lab. So I think all of this has been interconnected.
Yeah, you really need that interdisciplinary approach and communication, right, between all of the stakeholders.
Yes,
yeah.
Collaboration with everyone.
Yes, which can sometimes be tricky. I'm curious too about this the evolution of these clinical trials over the years. You've been involved in this research for so long. What have we really learned about how to do these clinical trials, or what have we learned from these clinical trials, perhaps?
So I think in the first trial we ran, which was an ampakine trial that was supposed to improve the connectivity in neurons in Fragile X based on the mechanism of what the Fragile X protein does. We did that trial with Rhonda Hagerman. She had just moved here to the Mind. And we learned that a lot of the measures we thought that people with Fragile X would be able to do, they simply couldn't do. And so that was an opportunity for us to throw out a whole bunch of measures because they weren't reliable, or they weren't doable, or there was some reason why they weren't good. And so we were able to start honing in on things that would be appropriate measures for trials. And I think as each trial has gone on, we've learned a lot. We've learned that the model for clinical trials at the FDA is that you pick one outcome and predict that that will improve. When you're going into FDA with a drug that worked in the mouse model for Fragile X, and it works for a lot of different phenotypes, a lot of the different manifestations in the mouse, and they tell you you have to pick the one behavior that's going to get better, that's a big problem, because you're looking to treat the whole disease and not one behavior, and you don't know which behavior to pick. And so as a result, we've picked the wrong behavior a couple times because we were stuck with this process that you would use to develop a drug for a symptom, not to develop a drug for an entire disease. And that's still, that's a problem that continues to this day, is how do we do that? And then we also learned a lot about social media, and that because social media was just becoming popular in our early trials, and we found out that sometimes parents post things online that can unblind the trial, or that can allow families to be able to rate a scale high enough to get in a trial, and then there's regression to their basic baseline, and then there's a lot of placebo effect from both of those things. So we've learned that we need to limit that. We've learned that sometimes something works in a mouse and the brain is just not set up in people anymore the way it was in mouse. Like over all those eons of evolution, certain receptors may not be as important in human brain as they are in the mouse brain, and so sometimes things won't translate. And so you need to have a very good marker of, okay, we see this thing improving in the mouse, but we also see the same thing improving in the humans, like an EEG change or a molecular change or a cellular change, something that you can say, yes, it looks like this mechanism is going to be important in humans as well as the mouse, and now we can directly translate it. So we've learned all those things. And we've also developed a huge number of better outcome measures than what we were using, 10 or 20 years ago. We have this new cognitive measure, the NIH Toolbox, that David Hessel has modified for intellectual disability and created standardization and manual, and that's one of the primary outcome in one of our trials now, and it's an outcome for cognition, which is the core problem in Fragile X. So finally we have something that's really directed at the core problem in Fragile X. And then we've done a lot of work on other outcome measures for other symptoms of Fragile X, and a lot of work on EEG biomarkers, that if we treat the patient with a medication and the EEG biomarker improves, that really shows we're doing something to the brain, and those measures are not really subject to much placebo effect, so we can actually see the brain changing. So I think we've made some really big advances, and hopefully they will be, now we are prepared to go into trials. Like if we had a gene therapy 20 years ago, we wouldn't have known how to do the trial. Now if we have a gene therapy, I think we're set up, we have good outcomes and we're set up to go. But we also have many small molecule therapies that directly impact Fragile X mechanisms. And I think other other disease fields like Angelman and Rett have watched what's going on in Fragile X and have learned that they have to develop these disease-specific outcome measures, and that they have to ask the community what's the most important problems to solve. And all of these kind of trial design issues are really interconnected in all the developmental disabilities. So anything we learn on Fragile X or anything we learn on Angelman syndrome is relevant to the other disease.
That's so great that it can be applied sort of across the board, and that you all have made so many advancements, right, in understanding how to measure these clinically significant factors, right, and perhaps influence their development. That's really, really fabulous. I am just so struck too by how much of an advocate you are for these communities. I think that is such a special connection that you and your team have with the people who are engaging in your clinical trials and coming to your clinics. And I'm just curious sort of how those reciprocal relationships have developed and how they've really influenced your research. And
yeah, I think these families, they have a hard time. I mean, they have a very difficult disease to deal with in many cases. In some cases, they're lucky and the individual in their family with Fragile X or Angelman syndrome is mild and easier to manage, but many of them deal with a lot. I mean, I have some patients whose behavior has been really challenging, and they've injured family members, and they've blocked themselves off from being in programs and so forth because of the aggression. And we work with these families constantly, with both behavioral methods and with psychopharmacology or medications to try to help behavior. And so you have so much contact with them that you get very close with them, and you really understand their issues and feel for them. And so they become, and if you're a good responder and get back to them when they need you, it really becomes a very close relationship. So you can't help but becoming close to these families. And you see each other at meetings and at different conferences, or if you go to Advocacy Day in Washington, you see the patients. So I think it just naturally evolves as a relationship with patients that you have a lot of contact with and are very close to. And this happens with all of our, specialty, diseases that are rare genetic diseases. I mean, these families, they don't have, it's not like it's diabetes where everybody knows what to do with it. These families have a very rare situation, and they may not have anybody near them who really knows anything about their management. So they're very grateful to have somebody that will actually try to help them. Even if we don't wind up solving their problem all that well, at least we're there to try to do what we can for them, and hopefully in the long run to get disease-targeted treatments FDA approved. That is the goal that we strive for and we fight for together. And we have some of our adult guys who've been in multiple of our trials, and they've learned, they know all of our coordinators going back 20 years, and they're super, they love to come to Rush, and so that's great because they're really helping us, with furthering the field and with getting to the point where we do hopefully have an FDA approved drug.
Yeah. Well, so I'm wondering if there's anything that you, or perhaps you think that these families would like the general population to know about Fragile X or some of these rare genetic disorders. Like if there was one thing that you wish more people knew, what might it be?
Wow, that's a little bit of a hard one. It's probably different things for different people, but I think the families would like the general population and possibly the FDA to appreciate just how big a struggle they have, like how many barriers there are to them like living everyday life because of this behavior or that behavior, or because they have an adult and there aren't enough job opportunities and there aren't enough programs, or the patient refuses to go out of the house. I mean, I think the struggle that we have in all these diseases, I don't think the general community recognizes just how big a struggle these families have.
Yeah. Well, thank you so much for sharing some of your work with us, and for just being such an amazing scientist and advocate for these families. It's been so wonderful chatting with you and learning more about your research.
Okay, great. Thank you.
Fabulous. Thank you. Well, that's all we have for today's episode of Science Minds. Thanks so much for joining us, and we'll see you next time.
The UC Davis Mind Institute was founded in 1998 with the promise to reduce and prevent the disabilities that can be associated with autism and other neurodevelopmental conditions. Every day, our clinicians and researchers make progress on that promise. Our groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other conditions associated with disability are helping affected individuals achieve their fullest potential. Please visit our website or our social media platforms to find out more about current studies, upcoming events, and how you can help make a difference.